Why is smell important? For starters, it is important for safety. If there is a gas leak, you can identify the gas leak by the smell of the gas that is leaking. It is important for aesthetic purposes like diagnosing whether you have body odor or not. It is important for diagnosis of cholesteatoma. It is also important for certain professionals like chefs, firefighters, winemakers, plumbers, sommeliers. It is an early sign of nasopharyngeal carcinoma, Alzheimer's disease, Parkinson's disease, multiple sclerosis, frontal meningiomas, etc. And the loss of smell is also important from a medico-legal standpoint. But where does perception of smell take place? Inside the nose? It takes place inside the olfactory neuroepithelium. This is an area which is not discernible to the naked eye. It is an area of pseudo-stratified columnar epithelium, different from stratified columnar epithelium. It is supported by a highly vascularized lamina propria. It occupies a small region of the nasal mucosa, about 2 square centimeters in area, in the upper recesses of the nasal chambers, lining the cribriform plate and parts of the superior turbinate, middle turbinate, and the nasal septum. Majority of the olfactory neuroepithelium lies inside the olfactory cleft. The olfactory cleft is a 1 millimeter wide opening that lies 7 centimeters deep to the anterior nasal opening, also known as nostrils. Only 10 to 15% of the inspired air reaches the olfactory neuroepithelium. So olfaction can be altered due to small polyps in the superior meatus without causing any symptoms of nasal obstruction, as majority of the air is still getting inspired without any difficulty. Olfaction, of course, means smell perception. But how is smell perceived? All land mammals, including human beings, have 4 major specialized neural systems for smell within the left nasal cavity as well as within the right nasal cavity. Now, the main olfactory system is the olfactory nerve, that is the first cranial nerve. The accessory olfactory system is rudimentary in human beings. This is the vomeronasal organ. It is rudimentary as well as non-functional in human beings. Then we have the trigeminal somatosensory system, which is mediated by the 5th cranial nerve. And finally, we have the nervus terminalis or terminal nerve, which is also known as cranial nerve 0. Now, the first cranial nerve, that is olfactory nerve, mediates common odor sensations. The 5th cranial nerve or the trigeminal nerve mediates both chemical as well as non-chemical stimuli in the form of somatosensory sensations like irritation, burning, cooling, tickling, touch, etc. Trigeminal nerve also induces reflexive responses like mucus secretions and halting of inhalation, which helps in prevention as well as in minimizing chemically induced or thermally induced damage to the lining mucosa of the nose as well as the lungs, because as you know, it's a one airway starting from nose to the lungs. Cranial nerve 0, as the name suggests, was discovered after the other 12 cranial nerves were already numbered. It is a well-conserved loose plexus of ganglionated nerves lying in close proximity to the vomeronasal organ. The neurons of cranial nerve 0 are reactive to the peptide hormone gonadotropin releasing hormone. So there must be an association with endocrine processes. However, it has not been proven so far. Now let's discuss the olfactory pathway. We'll discuss it in the following steps. The first step is odorants enter the nasal cavity either actively by sniffing or passively by diffusion. Then the odorants reach the olfactory cleft. Then the odorants pass from air phase to aqueous phase in the olfactory mucus. Then the odorants will reach the olfactory receptors. Signal transduction takes place at the olfactory receptors, where chemical energy is converted to electrical energy by a complex cascade of events, some involving activation of G proteins and various second messenger enzymes. Sixth step is olfactory bulb sends the signals to various parts of the brain. This is known as second order projections. And the final step is third order projections, when the signals are transmitted from one part of the brain to other parts of the brain. Now let's discuss the olfactory receptors. Olfactory neuroepithelium has 6 different cells based on the differences in morphology, differences in biochemistry, and differences in functionality. The first olfactory receptor cell, that is the most common, is the bipolar sensory receptor neuron, which is derived from the olfactory placode. It is of central nervous system origin and it extends odorant receptor containing cilia into the nasal mucosa. Then we have the supporting or sustentacular cells, which insulate bipolar cells from each other. It also regulates the production of mucus and also helps in transporting molecules across the epithelium and helps in detoxification and degradation of odorants. Number 3 is the duct cell of Bowman's glands, which secretes most of the mucus in the nasal cavity. Number 4 is the microvillar cells, which are poorly understood and they send tufts of microvilli into the nasal mucus. Number 5 is the horizontal or dark basal cells, which are one of two main classes of stem cells in the basement membrane of the epithelium. Next up, we have the globose or light basal cells, which are multipotent basal cells that can give rise to neuronal as well as non-neuronal cells, including horizontal basal cells. Now let's discuss a few key attributes of olfactory receptor cells. Olfactory receptor cells exceed in number every other sensory system except vision. Collective surface area of olfactory cilia is 20 square centimeters. Almost 6 million olfactory receptor axons coalesce into 30 to 50 olfactory fascicles, which are known as olfactory phyla. Olfactory phyla traverse the cribriform plate and pia mater to synapse with second order neurons in the glomeruli of the olfactory bulb. Glutamate is the neurotransmitter that excites olfactory bulb neurons, whereas dopamine modulates olfactory nerve input apparently. Olfactory bulb and its central projections also need to be discussed. Let's discuss them now. Olfactory bulbs are typically located on the ventral aspect of the frontal lobe on each side, just above the cribriform plate. The first synapse is between incoming bipolar olfactory receptor cell neurons and the spherical cells, which make up a distinct layer inside the olfactory bulb. These spherical structures are known as glomeruli. Each receptor cell projects to a particular glomerulus, and one glomerulus receives receptors from a restricted region of olfactory neuroepithelium. Considerable convergence of information takes place at the level of the glomeruli. The main afferent second order neurons are called mitral cells and tufted cells. Mitral and tufted cells send collaterals that in turn synapse within the periglomerular and external plexiform layers, resulting in reverberating circuits that result in both positive as well as negative feedback. Apical dendrites of mitral and tufted cells are influenced by interneurons and centrifugal fibers, most of which are GABAergic or dopaminergic. Apical dendrites, which are primary, they reach the secondary cortex, that is the orbitofrontal cortex, which is responsible for conscious perception of smell. Some cortical projections also relay through the thalamus. Some cortical projections reach the contralateral side also through the anterior commissure, largely from the pyramidal cells of anterior olfactory nucleus. So higher order brain regions targeted by mitral and tufted cells of olfactory bulb are as follows. Number 1, anterior olfactory nucleus. Number 2, piriform cortex. Number 3, olfactory tubercle. Number 4, entorhinal area. Number 5, amygdaloid cortex. And number 6, corticomedial nuclear group of amygdala. Third order projections occur in the brain in reciprocal fashion to mediodorsal nucleus of thalamus, posterior and medial hypothalamus, hippocampus, and finally to the orbitofrontal cortex as well, which also receives second order projections. Now let's briefly discuss the different abnormalities of smell function, the various terms associated with it, and what do they mean. First up, we have anosmia, which is the inability to smell anything. Then we have partial anosmia, which is the inability to smell some things. Then we have hyposmia, which is the decreased perception of smell, followed by hyperosmia, which is the increased perception of smell. Then we have dysosmia, which is the perverted perception of smell. Good smell is perceived as bad and vice versa. We have phantosmia, which is olfactory hallucination, olfactory agnosia, which is forgetting a smell. Finally, we have osmophobia, which is the fear of smell.